You often don't hear biology and music being connected together. Biology goes into the science category and music goes into the art category. When do you hear a scientist discuss the musical research he or she is doing? When do you hear the musician speak about the scientific impact his or her music is having on the world? Well, you probably mostly don't. I'm here to enlighten you about the connections between biology and music. There have in fact been studies conducted to understand the biological effect that music has on cells. This presentation will connect music to cell biology. I will be briefly going over two studies that present evidence on how music affects cells in hopes that you can see the important connection between the two. Here are some questions to think about throughout the presentation. Does music have an effect on cell division? Does music have an effect on cell growth? Why should music and cells be studied together? Before getting into the details of the studies, I'm going to briefly go over the cell cycle progression with you. S phase is where DNA replication occurs. G2 phase is a gap phase during which cells can grow. M phase is mitosis, where chromosomes are separated into two separate sets of chromosomes and end up in two nuclei. G0 is where the cells can exit from the cell cycle into a resting phase. G1 is another gap phase that occurs before DNA synthesis. The first study is titled Direct Effects of Music in Non-Auditory Cells in Culture. The study was conducted on a human breast cancer cell line, MCF7. The study looked at the response that MCF7 would have to music. The cells were exposed to 30 minutes of 3 compositions. 4 speakers symmetrically surrounded the petri dishes suspended on a platform as shown here. There were 2 controls in this experiment. There was a negative control where the cells were exposed to silence, and there was another control where the speakers were plugged in with no sound. This was to take into account the effects of background noise of the magnetic field that the speakers were producing. The cells were analyzed using flow cytometry. This slide lists the procedures that were used. The cells were tested 48 hours after being exposed to music or silence. They were washed in phosphate buffered saline, 5-minute centrifugation, resuspended, and stained with iodide. Here is a picture of a flow cytometer and another picture of how it works. The songs that the cells were exposed to were Mozart's Sonata for 2 pianos in D major, Movement 1. This was chosen because of what is called the Mozart effect, which is the belief that Mozart's music may induce a short-term improvement on the performance of certain kinds of mental tasks. The next song that was chosen by the researchers was Beethoven's Fifth Symphony, Movement 1, because it is one of the most heard compositions around the world. The final composition chosen was Ligeti's Atmospheres Movement 1 because it avoids conventional melody, harmony, and rhythm in favor of what is called as sound masses. As you can clearly hear from these examples, each song has its own varying different characteristics that make each piece very different and can affect the cells in very different ways. The results showed that both controls had a typical histogram with 2 peaks for G0, G1, and G2M, depicted by the arrows. The cells exposed to music showed a different result. The cells were more concentrated in S phase, and the number of cells in G2M were less. The important thing to note from this figure is that cells appeared in G0, which is the resting phase of the cell cycle. These cells are not dividing. This could mean that there is an increase in cell death. To test the cell death hypothesis, cell number and viability were measured. It was found that Beethoven's Fifth Symphony and Ligeti's Atmosphere resulted in lower cell number, but Mozart's Sonata did not change cell viability. Mozart's Sonata had similar cell viability as the speaker control. From this experiment, we can conclude that music does alter the cell cycle. Specifically, there was an increase in time spent in S phase and a decrease in G2M. There was an increase in number of cells in G0, which might mean that there was more cell death. Further analysis needs to be done because this was not statistically significant. There was a decrease in cell viability for the Beethoven and Ligeti music. A hypothesis for why this happened is that these two pieces have in common being played by an orchestra, but Mozart's was played by two pianos, having a lower frequency. It is important to note the need for controls in experiments like this. There are many variables that can affect cell growth based on different vibrations and other factors. The second experiment investigated the effects that sound would have on E. coli growth. E. coli was inoculated on Na and Nb medium. Na was for indirect viable count and Nb was for direct viable count. Na was incubated for 37 degrees 24 hours. Nb was incubated for 2 hours at 21 degrees Celsius. The sound treatment was in an acoustic chamber as depicted here with varying tonal frequencies, 1, 5, and 15 kilohertz. Results of the direct viable count showed that there was an increase in viable cells with the acoustic chamber versus the control that had no music. There was an increase in the number of cells. 5 kilohertz had the greatest increase in percent of cells compared to the 1 kilohertz and 15 kilohertz. Indirect viable count was measured using colony-forming units. Once sound was added, there was an increase in CFUs. It is to be noted that 5 kHz had a higher value than both 1 and 15 kHz again. Further experiments need to be conducted to understand exactly why. However, 5 kHz seems to be the optimal frequency for growth compared to 1 and 15 kHz. This image shows the indirect viable counts. Clearly, you can visually see that 5 kHz had the most E. coli growth. The 1 kHz had about the same as the control. 1 kHz was not enough to create any significant difference compared to the control. In conclusion, 5 kHz was the most significant in terms of cell growth. Control was also important in this experiment, and the researchers noted that they did have consistent findings with other researchers doing similar studies. Further research is clearly needed to make any strong conclusions. We can say based on these experiments that music does indeed have an effect on cells. Depending on the song played and the frequency of the sound, there are different results. This is significant because of the possibilities of music being used in therapy, development, medicine, stress, and more. Therefore, music should not be disregarded in the scientific community. As you view the credits, the music in the background will be Mozart's Sonata for 2 pianos in D major. Remember, this sonata was the only composition that did not have loss of cell viability when the cells were exposed to it. Thank you for listening to this presentation, and I hope that you won't forget the importance of music in the scientific community.